Thank you very much for inviting me to give this talk. I'm really excited. And so yes, I'm going to talk to you a little bit about some of the work that we've been doing at UKCEH around deriving indicators of biodiversity change from citizen science data, and some of the work that we're doing now to make some of this work, the outputs, the research outputs available for end users. So, as we all know, globally biodiversity is declining, and this is true for the UK as well. According to the latest State of Nature report, more than 40% of UK species have seen their populations decline, and we find wildlife in fewer and fewer places. Now, in order to bend this curve of biodiversity loss, we need not only increased conservation effort, but also a fundamental change in our production and consumption patterns. And the UK governments have responded to this crisis with a series of policies that set out different strategies for nature recovery. Now, at the heart of the success of all of these policies is monitoring biodiversity. We need to monitor biodiversity because we need to establish a baseline, and we need to track our progress towards reaching biodiversity targets and biodiversity goals. And that is because if we are not reaching those targets, then we need to rethink what we're doing. Unfortunately, monitoring biodiversity is really tricky. It takes a lot of time, it takes a lot of people, and it takes a lot of money. The Biological Record Centre, that is part of UKCEH, has been a leader and a pioneer in monitoring biodiversity through citizen science for over 57 years now. We work with a number of recording schemes and societies that are mostly volunteer-led organizations, and we collate all these species records from these multiple sources. We also work with age-structured monitoring schemes that collect the species records as part of a standardized monitoring protocol, which we call the structured data, as opposed to the national recording schemes data, which we call the unstructured. And thanks to all of these collaborations, we've been able to produce a lot of different outputs that allow us to understand how biodiversity is doing in this country. And this has included the publication of 120 distribution atlases that cover over 10,000 species, as well as 5,300 species trends that give us a view of how biodiversity has changed through time. So these data sets that we work with are for a variety of taxonomic groups, from plants all the way to different types of vertebrates. And this has been going on for a really long time. We have very long time series for some of these taxonomic groups, because some of the schemes have been active since the 1960s. And also it involves a huge number of people, particularly the thousands of volunteers that collect the data every year. Now, I don't have to tell you that this data is an amazing resource to understand how biodiversity is changing in the UK. But because of the nature of the observation process, which is usually opportunistic in nature, there is some unevenness in the way that the data is collected that, if not accounted for in the analysis, could lead to bias metrics of biodiversity. Now, when we talk about bias in citizen science data, we usually talk about bias in space, for example. So what you see in this map on your left is the distribution of records for one species of dragonfly across the UK for 1 year of recording. And you can see how this distribution is really patchy. So some areas are more covered than others, and there are a lot of big gaps. The coverage is not great. Once we stack up these maps across all of the species and across all of the years, we get something like the map on your right, which represents a period of about 10 years. So what this map is showing you is that there's great spatial coverage. There's literally only a handful of sites that have never been visited. These are all 1 kilometer sites. But what you can see from the color of these dots is that there is huge variation in the recording effort. So some of these sites have only been visited once during the 10-year period, and some of them have received more than 1,000 visits. So we need to take into account this huge variation in recording effort in our analysis. The other type of bias that is very often that we often find in citizen science data is bias in time. So this graph is a bit outdated now, but it's still valid. It shows you the number of records for all of these different taxonomic groups over time. And you can see that this number of records increases exponentially for all of these groups. Obviously, if this is really down to just citizen science activities just becoming much more popular, and so more recorders joining in these national recording schemes. And disregarding this trend in the data could lead us to erroneously conclude that biodiversity is increasing everywhere, when really it's just recording effort that is increasing. Also, we need to talk about effort and detectability. So effort is really important because there is a relationship between the sampling effort and the probability of detecting a species. So the longer we spend searching for species, the more species we're likely to find until it reaches a plateau. Now, we have no information on the effort that went into every one of these surveys that produced data. And we also know that the majority of records that we get are what are called incidental records. So only one species was recorded. Now, that does not mean that only one species was observed, and it definitely does not mean that only one species was present at the site at the moment of the visit. It's in some other records are we know that multiple species have been observed. Usually there is a bias towards species that are interesting in some way. Maybe they're particularly charismatic, colorful, or maybe they are rare species. And actually common species are usually underreported. So this unevenness again in the effort that is put into the different surveys makes these records very difficult to compare in space and time. And it makes it really difficult to infer absences from non-detections. We can't really tell that because a species was not recorded, it wasn't there. So over the years, we've been working to develop a reproducible workflow to derive robust metrics of biodiversity change from these biological records data. And we literally just published this paper that explains the whole workflow that we use to produce different types of outputs from this data. Now, as you can see in the figure on the right, there's a lot of different steps that go all the way from the raw data all the way to disseminating the outputs. So it really starts with the volunteer recorders who go out and collect the data every day. And then the species records are verified by taxon experts. After that, we need to look into some of the biases that might be present in the data. So we do a sort of risk of bias assessment to assess the spatial, temporal, and taxonomic representativeness of the data. After that, there's a data manipulation step where we try and homogenize this very heterogeneous data set by, for example, resolving taxonomy to species level only, and also only retaining records that are at a certain resolution, temporal or spatial resolution. And this manipulated data, this clean data, then feeds into the modeling step, where we then derive the species trends and the uncertainty associated with that trend. And then we evaluate the model by extracting some metrics from the models, but also by doing some expert consultation. We maintain a very good relationship with the national recording schemes and societies, and we kind of try and sense check the output from our models with the experts. Then it's only a matter of really producing these, aggregating all these results to produce metrics of biodiversity change, and for different types of applications, including biodiversity indicators, and then disseminating the results. In the next few slides, I'm going to just give you details about some of the key steps in this workflow. So we talked a little bit about some of the biases that we might find in citizen science data. But how do we assess quantitatively and also in a standardized way what those biases are and how they can impact on our metrics, on our metrics? Now, other disciplines such as medicine, for example, are a lot ahead of us in ecology, and they've been using standardized risk of bias assessments for a long time. So ecology is lagging a bit behind. So that's why my colleagues produced this tool for assessing the risk of bias in studies of temporal trends in ecology that is called the ROBIT. So as you can see from this graph, it really starts with defining your statistical target population, which means what is the thing that you're interested in saying something about. Are you interested in one species over the national territory, for example, over the whole country, or are you interested in one single population in one site? So defining what you're interested in, and then defining your inferential goal. What is the question that you're trying to answer with your data? And then it's all about going through different steps to describe the data and the data manipulation that you do and the data cleaning process, as well as understanding the different geographic, environmental, taxonomic, and other biases. And what you see here on the right is the output from a piece of software that my colleagues developed called OCASS, which is a now package that helps us to derive and extract these heuristics and summary statistics from the data sets. They show us basically how the data may be biased in space and time. So, for example, in this first plot, you can see an index that shows you how far away the data set is from a random distribution in space and time, as well as showing you, for example, the recording effort through time in space. So these sorts of explorations of the data allow us to really think about the patterns in the data and how those patterns, together with the methodology that we want to use to analyze the data, then will impact the output. So armed with this knowledge of the biases in the data, we can understand the limitations of our data sets. And by understanding those limitations, we can understand the limitations of our ultimate outputs that we derive from these data. So this leads us into our modeling step. So this paper that you see here was published quite a few years ago now, but it was a really important piece of work for us. This was published by my colleagues in 2014, and the aim was to test how effective the different statistical methods are in producing robust trends of biodiversity change from the sort of citizen science data that we use. So they designed some simulations and different sort of scenarios of bias, which is what you see in these different panel plots. And then they applied all of these different methods here to that data set, and then compared the performance of different models using a type 1 error rate, where type 1 error is the model able to detect a trend in the data when a trend exists in the data. So what you can see here is that the method on the far right, the one in pink, is really good at dealing with the biases in the data in all of the different scenarios of bias and keeping that type 1 error rate really low compared to a lot of the other methods here. Now, it can do that because it can reverse engineer the observation process by explicitly accounting for some of the biases that we know are present in the data, so that we can make accurate ecological inference and produce outputs that are unbiased. This model, this type of model, is an occupancy detection model. And the way it reverse engineers the observation process is by actually separating out the two processes into sub-models that are linked by the data. So we've got an ecological model, which in this graph is shown here in pink, where you're basically modeling the true presence, absence, or abundance of a species as a function of different environmental covariates, such as, for example, climate or habitat type. We can also have the second part of this model is this one in blue, which is the observation submodel. And here we are really modeling the probability of that the observer will observe and record a species as a function of different variables. So this means that in this submodel, we can include a lot of variables that might explain some of the biases in the observation process. For example, a proxy variable for effort, or some term that explains spatial bias, as well as things such as seasonality, for example. So this model is what we apply to a lot of the data sets, particularly the unstructured data. But what happens sometimes is that we are lucky enough to have multiple data sets available to do a particular analysis and answer a particular question. So we know that increasingly there's a lot of information that comes out of citizen science programs, and there's quite a lot of them. There's also some data that comes from structure surveys, from the sort of standardized monitoring schemes. But there's also a lot of novel ways of collecting species data, including environmental DNA, camera traps, and acoustic sensors. So what do we do when we have multiple data sets available to answer a specific question? There are multiple approaches that one can take. So the simple one is to model each of them separately with a separate model. That leaves us with a very tricky task at the end of trying to reconcile the results from the different analyses and the different data sets. Very often the data sets can give you very different answers, and then you're trying to figure out why they're giving you different answers. The other option is to just pick one and run with it. In this case, we would usually pick the one with the higher information content. So in this case, we've got 3 data sets, a presence-only data set, a presence-absence, and an abundance data set. We will probably go with the abundance one because it contains the most information. The problem with this is that it discards a lot of the information available. These 2 data sets are not going to be able to contribute to the analysis. Another way to do it is by merging the data sets together and model them as they were one. In this case, we're using all of the data sets. However, we are still discarding quite a lot of the information because we cannot derive count data from presence only. So usually what you need to do to merge the data sets together is to degrade the higher information content data to the lowest common denominator. So in this case, the count data become presence only data. So we are still losing a lot of the information. And also what this method doesn't allow you to do is to account for the different ways in which these data sets were collected. So if the presence only data set was collected through opportunistic sampling, for example, while the count data set was collected using a structured and standardized monitoring approach, you will not be able to account for those differences in the analysis. So the better way to do it is using model-based data integration. What that means is that we keep the data set separate. We model each of them using their own separate observation model that will describe the way that that data set was collected, and so the specific biases that might be present in the data. And then all of these observation models then inform the same ecological state model that is going to explain the actual presence absence of a species or the abundance of a species. And so we can derive one unified inference or one unified story from all of the available data sources that are out there. There are a lot of advantages of using this approach. It increases the quantity of data available because we're using all of the data sources available. We can also make the most of all of the data that is available because we are able to utilize the strengths from each of these different data sets. For example, opportunistic data is great because there's a lot of it, and there's usually very good spatial coverage as well as temporal spread. Structured data are usually smaller data sets, but they are very reliable, and they were collected using a standardized methodology. So we can borrow strength from both data sources. We can also reduce the uncertainty in the estimates and produce more precise estimates of species distribution or abundance or whatever your quantity of interest is. And it does this as a combination of increasing the sample size because we have more data, but also by partially accounting for some of the biases that are in the different data sets. We can also increase the extent of inference. For example, we can include citizen science records with historic museum records, as was done by my colleague in this paper. So we can extend time series in the past. And we can also translate between different ecological currencies because we can model together presence-only data with count data, which is something that we couldn't do in any other framework. So if you want to know more about this topic of integrated modeling, you can look at this user guide that we published last year with JNCC, which is an introduction, a non-technical introduction to model-based data integration for biodiversity assessments. It explains some of the advantages and also the methodology, and also presents a couple of case studies that apply this particular modeling framework. So where are we? We started with the raw data, and what you see in this cube is different layers that are basically the records for different species. On the x-axis is time, and on the y-axis is space. And you can see there's quite a lot of heterogeneity in the resolution of the records in time and space. So usually towards the more recent years, we have usually the records at a resolution of 1 kilometer and at a precision of 1 day in terms of time. But as we go back, we get data at a lower, at a coarser resolution, both spatially and in time. The data manipulation step makes that data set more homogeneous by discarding all of the data that are at a coarser resolution. We usually only retain the records that are at 1 kilometer resolution and day precision. And then we've done the modeling. So now we've got some model estimate for each species for each year and for different regions. So we've aggregated the space sort of variable to a region, where a region usually means country, so either a UK level or England, but we have been producing some outputs at a smaller sort of spatial scale. So what it looks like is this. So on the top here, you can see the trends in occupancy for different species of dragonflies in this case. So occupancy is the proportion of sites that are occupied by a species in the area of interest. So in this case is Great Britain. And so you can see here that the species on the left is a very rare species that was basically absent from the country for most of the time period and then appearing in the later years. There's another in the middle, another sort of quite rare species, but has a very stable distribution over time. And on the right here, there is a very common species present at the beginning in about 60% of the sites, and whose distribution is also increasing over time. So what we usually do is aggregate these indicators, these species-specific metrics into multi-species indicators. So in this case, we basically have an average across all of the species for this taxonomic group, which is what you see here at the bottom of this figure. So this is how species of dragonflies are doing in the UK between 1970 and 2020. So you can see a distribution that increases over time. And we can also derive metrics such as the proportion of species that are increasing, decreasing, or staying the same. So these are the outputs that come out of the models, and there are a lot of different applications that we've been able to use this workflow and these outputs in. This is the workflow that we use to produce the official UK Biodiversity Indicators. And what you see here is indicator D1C, which is the indicator for the distribution of pollinating insects in the UK. It is based on occupancy models and biological records data for 398 bees and hoverfly species. And the data comes from the Bees, Wasps and Ants Recording Society and the Hoverfly Recording Scheme. So very worryingly, as you can see here, the pollinating insects have shown a decline in their distribution over the time period, quite a significant decline as well. And you can see here that there is a really large proportion of species that are still declining over the short term, which is the last 5 years. Also, this is the same workflow that we use to produce the metrics that go into the State of Nature report, which is the most comprehensive assessment of the state of biodiversity in the UK. What you see in this plot is the occupancy indicator that looks at changes in species distribution. It includes a number of taxonomic group for invertebrate taxonomic groups, and also bryophytes and lichens. And as you can see, again, worryingly, this indicator is declining over that time period, showing us that biodiversity is not doing very well in this country. We've also started using this workflow to look at some of the drivers that might be behind these biodiversity changes. This was a paper from a few years ago now from my colleagues that looked at the impact of neonicotinoids on population changes in wild bees in England. And what you see here in these plots, you see in red is the trend in occupancy that was estimated by the model. And in blue is what that trend would have been like if neonicotinoid had zero impact on the bees. So what you see is that for these first two species, the Andrina and the Lasioglossum, which are species that are known to forage on crops that are treated with these insecticides, these species, the exposure to neonicotinoids has had quite a big impact on these species trends by exacerbating their decline by about 20%. On the other side, this Osmia species does not feed on crops that were treated with neonicotinoids, and therefore we haven't found much of an effect of neonicotinoids on this species. So they were able to find this mechanistic link as well. We've also started using the workflow to look at the effectiveness of some of the management in conservation. This paper was published in this year, in the last few months, and it looks at the effectiveness of protected areas in Great Britain, conserving the diversity of some invertebrate groups. And what you can see from the plot on the left is that protected areas do support higher diversity. So there's more species richness is higher in protected areas compared to the rest of the country. However, when we looked at the trends of those species, actually species are declining at the same rate inside protected areas and outside of protected areas. So protected areas are not being very effective at halting and bending that curve of biodiversity loss. So the workflow is in itself an output, and while we were developing this workflow, we also developed a lot of different types of outputs. This has included software. For example, the R package Sparta that we use to run the occupancy models that I showed you before. There is the OCSS package that I also showed you before that derives those metrics of bias in the data, as well as different other pieces of software that help us make this different steps in this workflow more reproducible. We've also produced data products, such as this data set of annual estimates of occupancy for thousands of species in the UK between 1970 and 2015, which is publicly available and free to download. And we believe that all of these outputs have great potential to contribute to the efforts for nature recovery by providing a more robust way to assess the state of nature and design the strategies, effective strategies for nature recovery. But there's there's big barriers to the uptake of all of these methods and research tools by the community of organizations and individuals that are tasked with the job of nature recovery on the ground. And some of these barriers are just the level of technical skills and knowledge that is required to use them, as well as just general lack of awareness that these tools even exist, and also difficulty in accessing them. So my knowledge exchange project, bringing the data revolution to nature recovery, aims to close this gap between the advances in data science and statistics that have been made by the research community over the last few years, and that community of organizations and individuals that need to make nature recovery happen on the ground. So there are three aims to this project. The first is to increase the stakeholder awareness and knowledge of the science that is available out there that matches their needs. And this will be achieved through a sort of consultation phase. We are right in the middle of that, well, towards the end of that consultation phase now, actually. So I've done a survey of the stakeholder group, as well as some interviews and workshops, and to try and understand what the needs of the stakeholder community are, so that then we can map those needs onto the science that is available. And then we'll identify some priorities for the next phase of development of the project. The second outcome is that data resources are used effectively to guide the efforts for nature recovery. And this will be achieved through the co-design and co-production of knowledge exchange products that will allow access, the stakeholders access to these tools and outputs. Now, these knowledge exchange products can be lots of different things, including user guides, such as the one that I showed you on integrated modeling, but also documentation on the software that we've already produced, more software, for example. But we can also use some of the digital solutions that are out there to make all of these outputs more accessible and usable. For example, we can use analysis notebooks such as this one that can integrate analysis scripts together with interpretation text and plots and figures that creates just a much more user-friendly interface for us to share our workflow and for users to actually use it and adapt it to their own data. We can also use graphic user interfaces, such as this Shiny app, which we designed to present our results to the experts. And this was designed so that the user could actually interact with all of these outputs and actually interrogate them in a way that it just wouldn't be possible with a static report, for example, or a static graph. Ultimately, I hope to establish a self-sustaining community of practice around the use of data science for nature recovery. And this will be achieved through testing the products produced in the previous phase to make sure that they're fit for purpose and also that they work in different contexts, disseminating and promoting these outputs, but also generating new ideas for future projects. And the impact that this could potentially have on my partner organizations could be huge. And here you can see it in my partner organization's own words. Now, there's a lot of information here, and there's lots of different points of views because there are lots of different organizations that are involved. But it really comes down to helping them effectively use their data to inform the management and efforts of for nature recovery. And it's about also allowing them to be more effective in communicating the biodiversity crisis to decision makers and also the general public, so that we can inspire the next generations to look after their environment a little bit better. If you want to know more about this project, you can go to the webpage. The link is down below. This explains a bit more about the science behind this project with some references to some of the papers that I talked about today, as well as what the partnership is like. And eventually it will include also some outputs. At the moment, it's just a blank page. And finally, I just wanted to thank NERC for funding this project and most of the research, to be honest, that I presented today. All of the recording schemes and societies and the volunteer recorders for generating such an amazing resource for us to use. And all of the partner organizations in the Knowledge Exchange Fellowship, because none of this would be possible without them. And thank you all very much for listening. I'm very happy to take any questions.